Hello, everyone. As you probably know, I'm Martine Walmsley. I'm Head of Research Strategy here at PSC Support, and I'm delighted to be joined today by Dr. Palak Trivedi. You're a member of our expert panel and a great friend to PSC Support, and many of the people on this webinar will have seen Dr. Trivedi at our information days. He's a consultant hepatologist at the Queen Elizabeth Hospital in Birmingham, and a world expert on PSC research, and you lead several important PSC research groups, Dr. Trivedi. So welcome. Thank you for giving up your lunch hour for us today.
Okay, thank you for having me.
And today we'll be talking about PSC research, and there was an international research meeting at the end of 2024. So I can think of no better person to update us on what we learned there and what's going on in the world of PSC research. As always, if you've got any questions, put them into the, I think it's a Q&A button at the bottom. We'll answer those as they come up. Or are you going to present anything at first, Dr. Trivedi?
No, I'm not going to present any slides, partly only because I did ask a few of the people who presented for their permission, and AASLD have now got a disclaimer saying that we have to ask their permission too.
Okay, so
I have not got that permission. I have not managed to obtain that permission, sadly, for this sort of public forum. So that's why I haven't, because Mark did very diligently ask me last week, and so I asked AASLD, but they have not replied. I'm afraid. So I can't present any material, but yes, we can certainly discuss what was.
We can talk about it.
Yeah.
Fantastic. Okay, then. So let's get started. I don't see any questions at the moment. Do you want to kick off with anything, or because I've got questions?
Yeah, sure. I mean, but if you wanted to start with your questions first, and then I can talk about some of the data that excited me, at least, from, and some of the presentations that excited me from the meeting that Mark and I attended.
Okay, great. Okay, so I'll ask first. The first one, it's a more general one, and it's because recently and so this meeting, we'll keep referring to it as AASLD or AASLD, and it's just stands for the American Association of Liver Disease.
American Association for Study of Liver Disease. Yeah,
so that's what we're talking about when we use that acronym. So yeah, some of the commercial clinical trials published bits of data here and there, and I wanted to ask you about endpoints, or how they measure how effective a drug is, because some of them seem to be quite good at reducing fibrosis markers, yet their main aim was to reduce alkaline phosphatase. Can you kind of help us understand that a little bit and why it's important?
Sure. So when Martine refers to fibrosis markers, these are either imaging scan markers or circulating markers in your bloodstream that reflect how scarred your liver is likely to be. We can't measure whether it's purely scarring within the liver or scarring around the tubes that drain bile, your bile ducts. But in essence, they're markers of scar tissue formation, scar tissue turnover, and in some cases, markers of scar tissue going away from the body. And a lot of the trials that are currently being conducted use these markers as a proxy that it's causing or inducing benefit, because if you've got less scar tissue within your liver, your liver damage and the degree of liver damage is slowing down, and it's less likely long term that you're going to run into problems from your PSC. And alkaline phosphatase is another marker in the bloodstream. It's part of the liver test panel that you will have at your clinic visits. But rather than representing scar tissue formation, it's a marker of bile duct inflammation, less commonly a marker of liver inflammation. And when your bile ducts are inflamed or angry, when you have an infection in those bile ducts, or if there's a blockage in the bile ducts that is progressing, that marker of alkaline phosphatase goes up. So there is a bit of a disconnect, because one is measuring scar tissue, the other one is measuring inflammation and blockages. So if you've got a particular drug that's being tested and currently under trial, and if the main goal of that trial is to reduce scar tissue formation, but it's actually affecting alkaline phosphatase, you're saying, well, that's a good thing because you don't want to have inflammation in your bile ducts, but is it actually fulfilling that proof of concept that the drug is working the way it's supposed to be working? In the grand scheme of things, it's important to see how long these trials are being conducted for, because if you have elevated alkaline phosphatase values, we already have a number of medications, antibiotics, so deoxycholic acid, and similar and other agents that improve bile duct flow, that in a proportion of people can improve alkaline phosphatase. But what you want to do is not just improve alkaline phosphatase in the short term, like these agents can, improve it, normalize it, and normalize it over a very long period of time, because we know in people who have normal alkaline phosphatase values long term, for not just days and weeks, but for months and years, it is incredibly unlikely that they will run into major problems with regards to their PSC. When it comes to scar tissue formation and markers of scar tissue, they take longer to improve, or longer to reach a point of stability in a clinical trial. So that is more likely to happen later on. That's what we see from a biological perspective. But if you've got a clinical trial that's telling you we've attained improvement in alkaline phosphatase values, we may not have improved your markers of fibrosis or scar tissue formation, but we've attained some degree of stability in it, then that's a good thing. And certainly two of the clinical trials that where some data was presented both showed that. They showed some improvement in alkaline phosphatase, and they showed stability in people who had who had received an active drug in a clinical trial, compared to the group of people who had the placebo, where markers of scar tissue formation went up and the alkaline phosphatase changes were not very impressive. And the two main studies that we're going to talk about in this context, one was the Integris trial, which used an agent called bexotegrast. That was a tablet compound administered to improve scar tissue formation. It's also currently in trials for another scarring disease called pulmonary fibrosis, so a lung condition where scarring happens like it happens in people with PSC in the bile ducts. The other trial was the SPRING trial, which used an agent called CM101, which was an injectable form given every few weeks of an anti-scar tissue formation drug. And both of these trials are phase 2 studies. So the main goal was to see that whether they're safe, but as a secondary objective, they showed that markers of scar tissue formation did not go up to the same degree as they did in the people who were in the placebo group across a range of different dosages. So both of these trials, one oral compound, one intravenous compound, both showing stability in markers of scar tissue formation compared to the placebo group. Yes, they were both safe, which is most important, and they also showed some reduction in alkaline phosphatase values over the weeks that they were studied. These are phase 2 trials. These are at the intermediate stage. They are now applying to the regulatory bodies in the United States and later on in Europe, in the European Union and the United Kingdom, where they would also be wanting to launch phase 3 trials. And those phase 3 trials, I'm hoping, will launch at some point in 2025. But unfortunately, the current age and day that we live in, it takes a number of months to get that regulatory approval, because there is unnecessary red tape for these companies to jump through. But it's very exciting data. And these are two of the first trials to show improvement in scar tissue formation. And we've not had data like this before from other PSC studies.
No, it's exciting, isn't it? And there's a couple of questions actually referring to CM101, which is the SPRING trial you were just talking about.
Absolutely. And before we just get onto those questions, the other good thing about this data, the one thing we're starting to learn, and for those of you who came to the Birmingham Information Day a few months earlier, we now have more data to show that itching in PSC is more likely depending on the severity of how bad your PSC is. And both of these agents, whilst they did not say only people with itch can take part, they both also showed that as well as fibrosis markers having a degree of stability, itch improved in people who took part and receiving the active compound. So that's another exciting thing for the PSC trial landscape.
It's interesting, isn't it? Because there's a study where it sets out to reduce itch. Is might be working the same way and also reducing fibrosis, but it might be easier to get market approval for changing a symptom. It's easier to show.
If we have a compound that improves the quality of life of participants, quality of life of people that take part in that study, then regulators are much more likely to say, well, this is affecting something that is much more meaningful on a day-to-day basis to people who are affected by it. And there are conditions that are much rarer than PSC that have got compounds approved because of their improvement in symptoms. It is much harder for a company, an investigator, to have a treatment approved for PSC based on improvement of a biomarker, even if that biomarker predicts long-term outcome with a reasonable degree of confidence. It is much more likely. So if we have a compound that improves a quality of life measure in PSC, the regulators are much more likely to give you a pass and a green tick rather than asking for more data.
Yeah, thank you. And that's why we're seeing these trials all designed in different ways. I think everyone's trying to get around it and show that their drug works in a way that is acceptable to these regulatory authorities. So the couple of questions we've got on the spring trial are, you've almost answered it already. What are your thoughts on the drug CM101? And the other question is, which trial drug is showing the most promise in your opinion? And then in next to it, CM101. There's quite a few going on at the moment, aren't there? So it's hard to answer.
Absolutely. So I'm very excited about the compound. We've done some work in animal models of CM101 here in Birmingham, and it's certainly got a lot of proof of concept data going behind it. The fact that CM101 shows what we call a dose response means that in the doses that have been trialed to date, the greater the dose, the more likely people are to respond to drug, and that tells you that the drug is actually working, it's doing something. So I'm very excited about it. My hesitation, and I have to say, and I'm going to say this with any compound under study, even the ones that I'm developing together with PSC support and other rare disease funders in the UK, I will always say there is going to be some hesitation and some limitation. That is just the way to be transparent. And you can't say everything is sort of a win-win without no limitation. My hesitation with it, it's an intravenous compound. So that's not to say people aren't going to be wanting to take part in the study. In inflammatory bowel disease, a lot of compounds we use are intravenous, and people
that means someone's getting it into their blood, aren't they? They're hooked up to a machine and it's going in through their blood.
Yeah, and they have to come to hospital to receive it. It's not something they can take day to day. But so that's one hesitation about it. When I say hesitation, that is one barrier compared to, say, for instance, an oral compound. So, but the fact that it's showing this dose response, the fact that preliminary data from this phase 2 trial is showing stability in markers of fibrosis and improvement in blood tests and potentially improvement in itch is very encouraging. So the other thing about CM101, it has not yet been trialed with other agents that people with PSC might be taking. So for example, many of you will have inflammatory bowel disease and be receiving other things for that, which you receive intravenously or take in tablet preparation. And the class of medications that's broadly called biologic therapy, so agents like infliximab, adalimumab, vedolizumab, or these small molecule sort of oral inhibitor, oral compounds, these next generation treatments like upadacitinib and so on, all these long for inflammatory bowel disease, they have not been trialed in combination with CM101 because those individuals would have been excluded from that study at the very start. There is increasingly a movement to help recruit those people into the next phase of CM101 studies, like in spring. But the challenge is we don't know if there's going to be an interaction. We don't know if there's going to be an interaction with these two agents. So that's another sort of caveat. The other thing about the this CM101 sort of trial is it needs to be studied, and there is some subgroup analysis in the existing data, but it needs to be studied in people where we know that their fibrosis is going to progress without intervention. If you're going to recruit people in who are very early stage and in the lifespan of the trial, you're not really going to see fibrosis progression, then there's no point studying the compound sort of in that way. The phase 2 trial recruited both groups, people where it's likely to progress and people where we're not really likely to see some progression. But the next iteration, I'm hoping, will enrich for people for participants who we are likely to see some progression, so we can really see if the drug works. You don't want to go and say, I've got a rock that's going to keep tigers away in England, where we're not going to see tigers on the street. You really want to, if you want to sell a magical rock to somebody, you want to go to a safari in Kenya or India or somewhere like that, where you can really test it in an at-risk population of people. And that's what the next iteration of trials is going to be. So that's my sort of take on it. I'm very excited about the data. It does have some caveats to it, and I'm hoping we can overcome those in the next iteration of trials.
It's good. So it's balance, isn't it, of getting the real world population, what is it like in the real world, but also making the study be able to tell you what actually the drug did and not something else, and making it safe, of course.
Yes.
It's complicated, isn't it? We always think it's easier than it is sitting at home, but I mean, there's a lot goes into designing clinical trials, isn't there?
And it's good that people are designing them and working in that space.
Yeah. Thank you. So let's get back to these questions, because there's quite a few here. So they're not all related to research that was announced at AASLD, but that's okay, isn't it, with you?
Absolutely fine.
So, does alkaline phosphatase increase if vitamin D levels are low and insufficient?
Yeah, so it absolutely does vary depending on what your vitamin D levels are. So ALP, or alkaline phosphatase, most of it is made from your liver and bile duct, your liver and biliary system. But it can also be made from other places like the bones. It can be released from the small bowel. It can also be released by the placenta if you're pregnant. So alkaline phosphatase, whilst it's part of the liver panel, it is also part of a bone panel in a different way. Now, as doctors and healthcare professionals, there are ways of telling us that it is more likely to be from your liver and biliary system, because your other liver tests, like your gamma GT, will also be elevated. But if that is normal and your alkaline phosphatase is up, it's less likely that it's going to be coming from your liver and biliary system, unless you've got some genetic reason why your other liver tests aren't going up. If vitamin D levels are low, then it is very likely that your alkaline phosphatase values are going to be increased, because your bone is turning over at a different rate. And there are other parts of the bone panel as well, like there's a hormone that sits in your neck next to your thyroid glands called parathyroid hormone, that's also released by that area, sorry. And if that is elevated, then then you will also have elevated alkaline phosphatase values. So in anybody who I see with PSC or other forms of liver condition, if they have an elevated alkaline phosphatase value, I do check vitamin D, and I do replace vitamin D if it's low, before thinking that all the alkaline phosphatase, all the ALP is down to liver and bile duct problems.
Thank you. And do you tell people, not people that are necessarily on trials or that have got high alkaline phosphatase, but your general PSC patient, do you ask them to take vitamin D?
I check it in people, and if it's low, I do ask them, and it's going to be it's low. It does fall if you've got a chronic condition, so that's why I check it. But it doesn't mean it's going to fall in everybody. But I certainly ask them to take supplements if they have low levels. I don't advise it in everybody. So if they've got normal levels, even in winter time where we don't get much sunlight, where vitamin D comes from, if they've got normal levels, I don't routinely ask them to take vitamin D.
Yeah, okay. I think it's something that people should check, though, isn't it? Because it can be the cause of fatigue in some cases.
Yeah, so it definitely contributes to fatigue and energy levels. There is data to show that if you're receiving treatments for any chronic condition, if your vitamin D levels are replete rather than depleted, then you're more likely to respond to that treatment. And I certainly do say people of dark skin, people like, so from a country of origin where I'm from India, as well as those from Africa and other parts where you've got darker skin as part of your heritage, it is more likely that you will have low vitamin D than if you're a fairer skin. So it is also important to have your vitamin D checked as part of routine health, and take supplements if it's low.
Good, good, thank you. Okay, next question. I'm going to just jump down a bit to talk about Nor-UDCA. This wasn't announced, but we all understand there's an imminent announcement about Nor-UDCA. Do you know when the results are coming? Have you got any, any indication? And what are your thoughts on this drug, if you're allowed to say?
So the Nor-UDCA trial, so just for those people who are not aware, ursodeoxycholic acid has been used for centuries in a variety of liver conditions, and it is licensed and approved for the use in the condition primary biliary cholangitis, PBC, which is different to PSC, but it's approved for that. There have been trials of ursodeoxycholic acid. So this, urso means bear in other languages, and it was what we call bear bile, and it's now manufactured synthetically. And it has a variety of things. It increases the alkali bicarbonate concentration in your bile. It makes your bile a bit more friendly to your bile ducts. It helps your bile flow easier. But sadly, clinical trials in PSC have shown that ursodeoxycholic acid does not improve the long-term life expectancy, reduce the need for liver transplantation. There are lots of asterisks to those trials from a bygone era, whether the right dose was used, whether they had enough participants. But from what we know now, urso does not improve long-term outcome, but it can improve liver blood tests. Part of the reason for Nor-UDCA being developed is a lot of ursodeoxycholic acid gets lost in the bowel, whereas Nor-UDCA is modified slightly. So the side chain of ursodeoxycholic acid in its molecular structure is slightly shortened. So all the goodness that urso brings, Nor-UDCA concentrates in the bile ducts and liver. A much smaller proportion is actually lost in the bowel when you look at Nor-ursodeoxycholic acid. There was a phase 2 clinical trial, so an intermediate phase trial completed many years ago, which showed in a dose-responsive way, Nor-ursodeoxycholic acid improved liver blood tests in people with PSC. So a large phase 3 trial looked to see if it improved blood tests, but also improved markers of scarring, fibrosis tissue, as well as had stability in markers of liver injury on a biopsy, not just scar tissue fibrosis, but also markers of bile duct inflammation, markers of bile salts being stuck within your liver. So quite a sort of a good marker of liver damage. So people in this trial had up to 3 liver biopsies at the start, middle, and end of this program. So it was a big exercise conducted throughout Europe, and we kept it open during the pandemic as well. The trial finished recruiting people 2 years ago, and the people who were recruited into that trial had to take part for a minimum of 2 years. So that the last person who was recruited would have finished their follow-up at some point in 2024. And when that follow-up data was collected, it now has to be analyzed. And so that's a big process. For a trial of that magnitude, the minimum 2-year follow-up period, as well as whatever 4-year follow-up period we've got for people who've carried on therapy into the beyond what was the minimum period, that is still being analysed, to my knowledge. And I've spoken to the sponsors of that trial. They are expecting some announcement of that from that trial in the first quarter of 2025. That's the most information I have from that study. In PBC, this other bile duct condition that I referenced, there was also a Nor-UDCA trial. That trial was terminated, unfortunately, at the phase 2 study because it did not show any additional benefit beyond ursodeoxycholic acid in that study. So that is not going to move forward in PBC, but PSC phase 3 trial has completed, and we eagerly await the outcome of that study to see if it improved liver biopsies as well as blood tests.
I do get goosebumps thinking about how far along the research has come, especially Nor-UDCA. And when you say it had a dose-dependent response, that means so the higher the dose of that medicine, the more response there was, or the more improvement.
The greater the improvement in blood tests were.
Yeah, so we will, like you said, eagerly await to see exactly what that drug is doing and how effective it is.
Yes.
Okay, next question is about the ELF blood test.
Huh?
Is the ELF blood test a more reliable method of measuring scar tissue in the liver? And I'm assuming compared to what we have at the moment.
Yeah, so L4 it stands for enhanced liver fibrosis, and it's a blood test. You know, we have it available, enhanced liver blood fibrosis test. It's available in the UK, and we don't check it routinely in your day-to-day clinic visits, but we do check it in people where things such as your FibroScans, that's an ultrasound machine that tells me the number of how scarred your liver is likely to be, rather than a picture, which is what the ultrasound gives me. So we do check it in people where the FibroScan value may be inaccurate, or we don't actually believe it. So if the reading is very high, but everything else tells me that it's probably not, there's probably not that much scarring, or conversely, if the value is really low, but there are other features of advanced disease, we sometimes use the ELF test in that situation. But it is widely used in almost all PSC clinical trials at present. When it says it's more reliable, I'm not sure whether that meant more reliable than alkaline phosphatase or FibroScan or a liver biopsy. It's certainly a more reliable marker than alkaline phosphatase. I wouldn't say it's more reliable than a FibroScan, but the variability from reading to reading. So if I was to do both tests, a FibroScan and an ELF test on somebody over a series of days, where I would expect to see some stability, ELF is more likely to be stable, whereas FibroScan is more likely to go up and down, you know, and have a lot more variation, where in fact there's not likely to be that much change in fibrosis on a day-to-day basis. And that's because FibroScan is done manually. And it depends on which direction you face the probe. It depends on whether people have eaten recently. It can depend on other things. Whereas the ELF test, the day-to-day variability is not seen. I wouldn't say that if you were to say compare a biopsy reading with a FibroScan on as well as a biopsy reading and an ELF test, I don't think that there is good enough data to say ELF is more accurate in that setting, but it's certainly less variable on a day-to-day basis compared to what a FibroScan is. And it's certainly more reliable than alkaline phosphatase at telling me scar tissue formation. And I personally believe it's a more reliable tool and has less limitations at predicting long-term outcome compared to, for example, alkaline phosphatase values are.
Thank you. And is it cheaper as well? That's because it's a blood test than fibrosis, a FibroScan or a liver biopsy?
It's certainly cheaper than a liver biopsy, because a liver biopsy requires a lot more personnel. So it will require an ultrasound a trained radiologist to do the biopsy. It will require the bed space time and the nursing capacity to look after you for the hours after the procedure, and the hospital bed time, and the pathology processing time and histopathologist time to look at a slide. Whereas an ELF test is a blood test that you just put into a machine. So you take the bloods, you post it, it's done in a machine. So it's definitely cheaper. It is not cheaper than a FibroScan though. So we as doctors don't get paid extra. The nurses don't get paid extra. We just do it in the clinic room.
Yeah.
Do a FibroScan. So we will be charged a maintenance fee. We will be charged a flat fee for purchasing a machine for our unit, and we can use it one time or 1,000 times. And there'll be a maintenance fee, like a subscription, that we would have to pay for those machines to be serviced and MOT'd every so often. I don't know what the interval is, but the more people that use a FibroScanner, the cheaper it becomes, because it's not like your car, where when you get to 10,000 miles you need to have a service. It's it's just a time period thing. So every year or so it will need to be serviced and maintained. So FibroScan is probably cheaper in a big volume unit, I would say.
Thank you. So I'll go back to some questions that are specifically about clinical trials.
Yeah.
And this one is about Rectify Pharma. They announced some positive results from their drug, and they were saying that it improves bile composition and enhances bile flow. What are your thoughts on this?
So I think it's very, very, preliminary data. It's not control trial data. It's mainly in most of their data is from say animal models and synthetic models. So we really need to see what a clinical trial looks like compared to a control group. So it's encouraging mechanism of action, but we need proper in-human clinical trial data compared to, say a placebo group before we can determine whether it has any long-lasting effect. But yes, it's a good mechanism of action. I suspect it will be useful as an adjunct compared to, in addition to some of the other compounds that we're currently developing.
And from a patient point of view, for me, it feels really positive that potential new trials are coming along, and we're not just relying on one or two, because they all seem to be doing slightly different things that might help us.
They do. Yeah, they do. And again, even the two that we started about at the beginning, if you have, I can imagine an age where you would hope both trials in a phase 3 program meet their primary outcome measure. They both get approved to some degree, and we might be putting people on two therapies because they gain a bit from one, and then you gain a bit from another. And you might put people on a bile acid type therapy to help bile flow, and an anti-scar tissue formation therapy to reduce the chances of further scar tissue developing based on what's already started. So not that I want to give more people lots of compounds and beyond more than what they need, but but you can imagine that sort of therapy with different people at different phases would be able to benefit from different therapies.
So basically, we might be looking at a personalized cocktail.
Yes, exactly.
For ourselves. Yeah.
Like we do with some other liver conditions. Yeah, absolutely.
It would be, good.
Yeah.
Okay. Thank you for that. Next question is about what are your thoughts on the new PSC trial on the treatment that has been a game changer for treating psoriasis? So this is the brodalumab trial, Saber.
Yes. Yeah.
What are your thoughts on that?
Yeah, so it's a very, very interesting proof of concept study. And this is for those of you who are not aware, interleukin 17 or IL-17 is a pro-inflammatory molecule that is made everywhere in the body by a certain type of immune cell, your T cells. And it has been shown in clinical trials, as alluded to, in the dermatology, in the skin space, to improve markers of psoriasis. Because this pathway is active in people with colitis, it's active in people with primary sclerosing cholangitis. Colleagues slightly north of where I am, up in Norwich, Norfolk Trust, are doing a proof of concept study without a placebo group. So for any of you interested to take part but are a bit shy of the placebo nature of certain trials, they can receive this therapy, brodalumab, and in an effort to improve their markers of inflammation. It is a proof of concept study, and if the safety data holds true, and there is some preliminary evidence that it improves, say alkaline phosphatase values or markers of scar tissue formation, or even MRI-based changes, then a randomized control trial would be needed next, compared to a placebo group, to see if it has any additional benefit. I know that some people have raised a concern when this compound was used in psoriasis and other areas, because people experienced a deterioration in mental health when they receive this particular type of compound. I'm not saying the same thing is going to happen in any way, otherwise I wouldn't be supporting the trial in that way, but that has been raised as a question. But when I've mentioned it, and some people have gone away to look at it, saying mental health worsened in people who were in certain trials of psoriasis. That can be for a variety of reasons, but in this particular trial, we are keeping a very close eye on it. To my knowledge, nobody who's taken part in it has reported that side effect, which is very, encouraging. I'm hoping that Birmingham will also open as a site soon. There was some requirement to have healthy volunteer scans performed in our MRI unit, but there was no participant information sheet for those healthy volunteers that was available. So if you're a healthy volunteer and you need to have a baseline scan in the MRI scanner, you would need information sheets saying if we pick up something incidental, how would that be acted upon? And that hasn't yet been made available to us in Birmingham, but I believe now that the team in Norfolk, Norwich are now putting an amendment to their protocol saying that that would be an optional thing rather than a mandatory thing for a site to open, because the type of MRI they're asking for is different to what we use in day-to-day practice. So then we can certainly open, and other units would then open.
Thank you. And if anybody wants to see how to take part, and it's on our website. If you just search for Saber PSC, so SAPR.
Yeah, please do,
And it is a concern, isn't it, if it's got the potential to lower mood, but people are very closely monitored on trials, so it's not something that you'd just be left on your own. It's you're very closely monitored and checked upon.
Yeah.
Okay, good. I'll click done on that question. The next question is not a trial related question. It's, do you believe PSC or autoimmune conditions are linked to trauma or stress?
I haven't. A lot of people ask this, this question, and it's important to query that. I haven't observed that people who have more traumatic backgrounds or have more stressful lifestyles develop autoimmune conditions with an increased frequency. But what we do observe is that people who have conditions diagnosed, if they have major traumas or stressful periods in their life, their conditions are more difficult to treat. Their conditions are more difficult to manage on a sort of on a visit to visit basis, and they are more prone to having certain flare-ups. Now, that might be because their own personal health concerns may not be as much of a priority as the traumas and stresses, other stressors that are going on in their life. That is entirely possible. We haven't observed, though, that people with more trauma experience more autoimmune conditions. And studies to this effect I mean, they're very old now. It's even difficult to find them online, but I remember learning about them in medical school. We never used to see such a flurry of autoimmune conditions after, say, Second World War, where a lot of people were under trauma and stress. There is a lot more compelling data showing the link between westernized diets and the epidemic that we're seeing in autoimmune conditions. And about 4%, it was the latest statistic that somebody reported to me, one of my colleagues in the States, John Vierling, who's researched autoimmune liver diseases twice as much as I have. Sort of he's sort of still practicing sort of 7 days a week at the age of 80 or 80 odd now, I think. And he has certainly sort of quoted that prevalence. And there certainly is an increase in in countries that are adopting a westernized diet with more processed foods and quicker preparation foods compared to what's going on. And you'll be surprised, even for those of you who buy, say, pre-prepared, say grated cheese, for example, because it's easy to use and convenient. They, in the UK at least, we have potato starch to prevent as an anti-caking agent. But in the United States, they use much more synthetic compounds for that. And there's been some data showing that if you grate your own cheese versus using pre-prepared grated cheese, there is a big difference in how the processing of that works, and that's a lot much more of an inflammatory food stuff that you're putting into your body. So I wouldn't say it's linked to. I wouldn't say the onset is linked to trauma or stress, but trauma and stress we've observed it can lead to more difficult to control, difficult to manage, and recurrent flare-ups of any autoimmune condition you have.
Yeah. I mean, we also hear a lot of stories about significant stress-related events before diagnoses, but I guess you'd need to speak to everyone that's had a stress-related event to see if they get an autoimmune disease to really understand. Good, thank you. Just because our purpose was really about the AASLD meeting. Are there any, I mean, we talked about the spring trial, which was CM101, and the Integris trial. We've also talked about Rectify. Were there any other announcements or studies that stood out to you in that meeting that you want to talk about?
Yeah, sure. So things that might be, I'll talk about things related to PSC in particular, and things that are not directly related to PSC, but a bit more indirect, that we should sort of, that are exciting because they have translational components to PSC. So another trial that is recently completed recruitment in the UK is of a bile acid compound called elafibranor. So elafibranor is a therapy that's licensed for the treatment of primary biliary cholangitis, PBC. The trial in PSC, the phase 2 trial, recently completed recruitment in the UK. And there was more data to support elafibranor, even in people who did not respond to ursodeoxycholic acid and other bile acid therapies in PBC. That was presented at the American Liver Meeting. And there was data to also show that it improved markers associated with long-term prognosis in PBC. So this encouraging data in a related bile duct disease has translational aspects for PSC. The other compound that is something that interrupts bile acid resorption in the body, so an ideal bile acid transporter inhibitor called volixibat, has some interim trial results in PBC were presented from the Vantage study in PBC, and they showed significant improvements in itch intensity in the primary biliary cholangitis program. And many of you would be aware that that trial of that same agent is currently recruiting in the UK in PSC. So the Vistus clinical trial, which Birmingham are taking part of, as well as several other sites, they are actively looking to improve itch in people with primary sclerosing cholangitis. And if you're itching and you haven't found a solution to your itch, I strongly encourage you to get in touch with us here in Birmingham or one of the other trial sites. You can see the list on the PSC support website to take part. But the fact that the Vantage trial showed itch reduction in PBC as an interim analysis was very encouraging. And whilst they haven't released any interim data yet in PSC, because it's slightly earlier in this process, the fact that we're actively recruiting and there's activity in the itch space gives us an opportunity to explore the effects further. So that was an exciting bit of data in the PSC space. The third thing I wanted to mention was some data on quantitative MRI scanning. So MRCP Plus, which was the scan that I referred to when talking about the Saber trial, the brodalumab, agent. This is the scan that is not available as routine clinical practice in the UK, but we do use it for a research tool. So this is a post-processing artificial intelligence way of quantifying what your bile duct volume is, what the bile flow is like, the number of strictures, narrowings that you have in your bile ducts, the quantity of dilation upstream of those narrowings. And it's a tool that Oxford have developed, and we use it as a research purpose. And there is data that the Oxford group, our group, Toronto, Milan, Paris, a whole variety of organizations have published on, showing that it picks up some bile duct changes before normal MRI scans can. And it can also predict long-term outcome in people with a reasonable degree of certainty. But what we don't know yet about this about MRCP Plus, whilst it gives us lots of pretty pictures, is whether, whilst it predicts these changes earlier, does it add value to what my radiologists can do? So if I compare both technologies, conventional MRI and artificial intelligence MRI, whilst the changes can be picked up earlier, and both my radiologist and the AI can tell me who's more likely to progress, does it offer an added advantage? Can it reclassify people that my radiologist tells me are not progressing to more of a progressive phenotype with a better degree of accuracy? And that's something we're really asking the company to help us do. And as yet, they've made encouraging noises, but they haven't yet supported that type of work to show that it gives us anything more than a pretty picture or an earlier signal. Does it offer added benefit beyond currently available technology? Because if it does, then all of us would want to use it in our day-to-day practice. But there was more data supporting the fact that it's got some prognostic utility in people with primary sclerosing cholangitis. So that's certainly something that I was encouraged by. And the last thing, there were lots of discussions, whilst it wasn't new data, there were lots of discussions around the use of oral vancomycin, which is by some of our colleagues in North America. And oral vancomycin is a gut-specific antibiotic that has been shown in several different studies to, yes, as an antibiotic, change what the gut microorganisms look like, but preliminary data, including a trial that we completed 2 years ago here in Birmingham that's now published, to improve markers of bowel inflammation, and in some people with bowel inflammation also their liver blood tests. And I'll be talking about that in the European liver meeting in May this year, about oral vancomycin and PSC. So there was lots of discussion about it, although not any new data in that space.
That's something that patients talk about a lot, is the vancomycin, for sure.
Yeah.
So we really want to see more research on that just confirms or doesn't confirm what we want it to do. And just going back to the MRCP plus, from our point of view, when we see that amazing 3D image that this scan can produce, it feels like we can understand what it's saying, because it will show us where there's a stricture, so where there's scarring in the liver and where it's deforming the bile ducts. And you're saying, like, it's showing bile duct changes over time. So if we're looking at it saying it had this many strictures, or this was a total volume of the bile ducts, and that changes, it feels like that should definitely mean something. So you're saying, yes, there are other tests that can tell that sort of thing, or an equivalent, but really we want to know, does the MRCP plus do it better, or does it give us some more or different information?
Exactly. So for those people who've got children, when you went to the to your sonography appointments, you would get a black and white grainy image of what baby would look like, and the sonographer would be able to show you the head, tell you what the sex of the baby is, show you some of the structures, but beyond the head and the body and maybe the limbs and a healthy heartbeat, there's not much more that you or I would be able to tell from that, unless you're the person doing the scan. But now you can pay privately and you can go and get a 3D image or a color image in a private sonography appointment. We now, we don't have any good data to show that private 3D scan of baby adds value to telling you, is your baby healthy? What the growth is like? Are all the structures okay? Than a normal, ultrasonographer doing your scan on the NHS. So what we need to now know is MRCP plus the same sort of principle? Is it just a slightly prettier, fancy, more interpretable, a more something that you can visualize better version of your MRCP plus, but actually it doesn't tell you anything more than what a radiologist writes in a report and what your clinician should tell you prognostically? Or is it something that actually is next generation, and we should all be doing it for all our MRIs, because it adds a much greater degree of value, irrespective of how much it costs? It's not a cost reason, which is why we're not adopting it in clinical practice. We just need to know that it means more, and the company needs to allow us to study it in that way.
Yeah, exactly. And when you say prognostic utility or prognosis, you mean how good is it at helping us understand what might happen to someone in the future?
Yes, exactly. Exactly right. Exactly right.
Okay, so we'll keep watching that and hoping for some more research on it.
Yeah.
Thank you. We've got quite a lot of questions here, so I'm trying to scan through to see if there are any particularly related to research. Oh, this is one I like asking you. When do you think we will get the first drug approved for all patients with PSC, not only just being able to take it through a trial? Do you think it will be within the next 2 years?
If we have one drug approved, the others will come in quite quickly, because we will have a pathway to approval. At the moment, one of the biggest barriers that we have is not we don't know why people get PSC, but we understand several mechanisms that lead to people's PSC progressing and leading to bad things. We can develop a wealth of compounds that can interrupt those axes. We can design trials. We can, as the person who asked the question, we can design studies to interrupt those pathways and show that those pathways are being affected in a positive way. But the biggest barrier that we have is the people who give us the green light saying, yes, you have approved therapy. They, at the moment in PSC, they tell different investigators and different sponsors for these studies, different companies, different things, saying in order for you to show that your drug works and for us to approve it, they will tell drug company A, this is what you need to show. And they will tell drug company B something not completely different, but relatively different. So the goalposts are not different, so are quite different from one to the next. And when and it's not as different as, okay, your cake needs to have this much sugar in it, and your cake needs to have this much sugar in it. And there's a difference in the quantity. It is very much like saying to you, right, my children can do something small and make my wife very happy. I can do the same thing and it makes no difference whatsoever, because the goalposts for me to do something are very different to my children. And it's that big of a difference as an analogy. And there was a long time where the regulator said you need to show stability or improvement on a liver biopsy. But we know that in people, unless they're very end stage or very early stage, the bit in between, the biopsies from one section of the liver can look very different to another part of the liver. So you would always get this, this rule of threes. A third of people in a trial would show improvement, a third would show no change, and a third would get worse. They've now changed that, and they've said that for the companies like Integris, so the trials like Integris and Spring, they have said you need to show a benefit on clinical events, so less frequency of cholangitis, a lower incidence of people progressing to the point that they develop cirrhosis, less people developing features of liver failure. So they are now changing that. But what do you classify as liver failure? What do you classify as a flare of cholangitis in somebody whose liver test may not change much, but they have pain and a fever and shivering and shaking and their urine changes color? These sorts of things are different. Different things are being told to different people. So it's a very challenging landscape at the moment, despite all the encouraging noises. I suspect what will happen is we will get preliminary or provisional approval of one compound, and then the others will swiftly follow through. Nor-UDCA is the front runner in that race because that's completed recruitment and follow-up in Europe. It hasn't run in North America, so in Europe at least. And so at least from a European perspective, I'm hopeful, and I can't be more optimistic than that, but I'm hopeful that we will get the first provisional approval it's sort of 12 months after Nor-UDCA announces a positive result. If Nor-UDCA announces a slightly in the middle of the road result where it's improved some things but not everything, then sadly we will still have to wait for the other phase 3 trials to start, recruit and complete, and it will be several more years, minimum 3 to 4, before we get a drug approved for some people with PSC. For all people with PSC, it really depends at what points they come through the door, and I think that will still likely be sort of 5 or 6 years minimum away.
Yeah, a drug can take 13 or 14 more years, 13 odd years to go from concept to being available to patients. They take such a long time, and that blew my mind when I first started looking at research. I thought it would be, there's a clinical trial, you've tested the drug, done. But it's not. There's all these different phases and have to be really carefully looked at, and like you said, tailored to what that drug may or may not be doing.
Correct.
And even when they get positive results, we need to still work with the authorities to make the drug available to patients, because it goes through like a cost analysis. So there's a long way to go, but we're not going to give up. And I would also say to people, if you're thinking about going on a trial, don't not bother, because we've got these potentially good results coming up. We still need to follow through on all these trials to know how they're going to help and what they may add to the solution, in inverted commas. Good. So maybe in the next 2 years, maybe not in the next 2 years, depending.
I think the earliest that we will get some sort of provisional approval in Europe, UK included, will be I think 2026 or maybe the end of 25. That's the most optimistic I can be at the moment. One thing I must say though is that we have compounds that are under investigations that are being repurposed, so things that are already approved for the treatment of other conditions that don't currently have a patent or a license. And whilst they may not be approved in inverted commas for the treatment of PSC, if we show that they have some benefit and we already have them available, we can already start to use them, like oral vancomycin, for example, like bezafibrate, which is currently sort of being used to lower, sort of lower cholesterol, and we use it for treatment of PBC. Trials of these agents are still ongoing, and we don't need a regulator to say, yes, you can use them here in the UK and Europe. In other countries like North America and so on, it's slightly more tricky because your health insurer has to approve you to go on that treatment. And that's a whole different ballgame. But sticking to Europe, UK and Europe, if we show that bezafibrate works or vancomycin works, because we have them off patent for treatment of other GI, gastrointestinal and liver conditions, we can start using them altogether. Now, I'm not evangelical about any of these treatments. They have to be evaluated properly. But I do use them currently in my day-to-day practice for people where there might be some benefit, in my clinical opinion.
Good, thank you. And I'm going to finish on one last question. It's about Fargo, because that's your baby.
Yeah.
When will you publish the results for the Fargo trial?
Yeah. So it cannot be earlier than summer of next year. And that's for two reasons. One is recruitment is still ongoing. The trial has not completely recruited yet. So if anybody's interested in fecal transplantation as a treatment for your primary sclerosing cholangitis, please get in touch. We're not the only site to recruit. Open, Oxford are open, London are open. Sadly, Norfolk and Norwich, which were going to be our sites, are no longer able to take part, I think, because they're doing Saber and they're recruiting to that. So if you want to take part in that, please do. But we are certainly sort of open, and we are looking for more and more people to take part. Once we have completely recruited, it's a year, 48 weeks plus or minus 4 weeks for people to complete their follow-up in that trial. And once they finish, you have to allow us about 6 months minimum to analyze all that body of data. So we are hoping to finish recruitment this calendar year, and we will then complete follow-up for those people by sort of next year, and then analyze results as quickest. So if we finish recruitment by the summer or the autumn, then then we can start analyzing results the moment that the last person in finishes their follow-up, which will be next summer or autumn, and hopefully by 2026 we'll have some good readouts for you.
Yeah, thank you. So if anybody, the Fargo trial, it aligns with when you've got your colonoscopy, doesn't it? So if someone's got their colonoscopy coming up and you want to go on a trial, look up the Fargo trial, because that could be perfect timing for you.
And there are lots of people who may have had a colonoscopy less than 12 months ago who have actually very clearly said they still want to take part. So they brought their next colonoscopy forward, and we will, that is a discussion. If it's just happened, your colonoscopy, then we will probably say it's not worthwhile. But if you're certainly sort of closer to your next one than you are to your most recent one, a lot of individuals have expressed interest in bringing their next one forward. And that's a dialogue for yourself and your local physician and the trial site to have. We can be flexible and open-minded, but the main thing is keeping safe. We don't want you to have too many invasive investigations too close together just for the purposes of a trial.
Yeah, and bowel prep's not that great, is it? So it's not.
But that's another thing.
I mean, you want every week.
Yeah, exactly, and things, and it is a phase 2 trial. But because we use fecal transplantation for the treatment of other things, if there is a positive signal, then it's something that we wouldn't need to jump through the same amount of hurdles before we can start offering it to people on a case-by-case basis.
Thank you. I could talk to you all afternoon on all these different things. And we've got tons of other questions, but I'm sorry to everyone that has asked a question and that we've not got through it. But Dr. Trivedi, thank you very much for giving up your lunchtime again for us. We really appreciate that. And before I sign off, our next Ask the Expert is Wednesday, the 12th of February at 7 o'clock with Dr. Ferguson from the Queen Elizabeth. And the liver transplant coordinator from Derriford, Katie Ramos, and some patients. So that's Keith Anderson, who many of you will know because he had his transplant back in 2003. and is very active on our groups, and also Mark, who's been in the background of this Ask the Expert, answering questions.
Hello.
He pops up. Hello. He'll be on that as the expert as well. So I think that'll be a really interesting one to do. So hope to see you there. And with that, we'll all go. Thanks very much, Dr. Trivedi, and thanks everyone for joining us. Take care. Bye-bye.